initial androgen responsive to here, then in clinical, we need to have more research going on. So this is one we published in 2015. How I try to explain to you, use our paper to show you how we perform the experiment, how we determine the functional role of tyrosine phosphorylation by ERB2 in regular androgen sensitive and castration resistant prostate cancer cell. So the castration resistant prostate cancer, to you, maybe the title is a little bit of confusing. So initial cancer we call, say, androgen sensitive, then become androgen, we can say androgen unresponsive or insensitive. That is the cell we show you, androgen can no longer stimulate cell growth. So the castration resistant prostate cancer cell is actually advanced prostate cancer. So even a patient under the castration, so in the clinical case, initially is androgen sensitive, so we can surgery it. If we did not surgery, we can use a radiation therapy. Then when the cancer come back, relapse, and the metastasis, then at that time, the gold standard of the treatment is androgen deprivation therapy. We remove the androgen. So either surgical castration, just remove the testes. And or we use a chemical castration. We give patient anti-androgen. Okay, so then the tumor will suppress. But then, unfortunately, all the chemotherapy only for a time period time. It's only for a short period of time. Maybe 1, 3, 5 years the most. They will relapse again. They will come back. The tumor will come back. Then we call that stage now, we call the castration-resistant prostate cancer. We call the castration-resistant prostate cancer because the cancer cell progress under the castration condition. There's no androgen under the castration condition. Okay, the low androgen condition. And it has a, when you check the literature, they have a terms called androgen insensitive or androgen refractory prostate cancer cell. But now we don't use those terms anymore. We use a castration resistant because for the hormone refractory is the same as they think under the androgen deprivation therapy, the hormone is cancer cell come back, then that's what they call hormone refractory. But in reality, the castration resistant prostate cancer cell still require androgen. It still requires androgen because we can further use a new generation of the anti-androgen. We can still suppress it. Okay, we can still suppress it. So there are many stages, but the bottom line is prostate cancer cell require androgen function, require it. It's just different kind of, they might be sensitive to a low amount of the androgen. So even a very residual androgen in the circulation castration stage, they can still grow. So how the cancer cell can respond to so low level of androgen in the circulation? It's signal transduction pathway actually provides additional growth stimulation. Yeah, androgen is required. It's essential for their survival. Then the signal transduction actually gives them additional cell growth. Okay, so this will be. So that's what we try to demonstrate how the androgen regulate. Oh, where is my slide? Where is my video? Okay, so how we determine ERB2 is involved in androgen stimulation and the castration resistant prostate cancer cell. So again, the first is we have two different prostate cancer cells. So LNCAP, as I mentioned earlier, is androgen sensitive. And we now have another cell called MDA-PCA2B cell. It's also express androgen receptor. You can see here, and they are also androgen responsive. But then interesting is we can just passage the cell in the cell culture. Then they will automatically, gradually become androgen independent. They can survive very well in the absence of androgen. So exactly as patient in our body, in the patient, exactly as the cancer cell in the patient. Early stage androgen sensitive, then following the time of progression, they become androgen independent, castration resistant. Okay, so inside the cell, you culture them, they change. So this is a take-home message for you guys. Culture the cell, be very careful. They do change. Don't think it's a cell line, they are immortalized there forever. They do change. Just depending on what kind of cell line, what is the change, that's all. So both LNCAP cell and the MDA cell, initially they are androgen sensitive, they express AR, androgen receptor. But then the C81 cell, we call the LNCAP81 or the MDA AI cell, they become androgen independent. So that means they can grow in the condition after we remove the androgen. And with the androgen, if we add the androgen, the growth stimulation is only marginal. So we use the two cell line, two independent system, and the two cell line. Then we try to compare what is the change. So we analyze a lot of molecular signal pathway. Okay, so for example, the major, we find the ERB2, the tyrosine phosphorylation at the position tyrosine 1221/2. So that means in the 1221 and the 22, both have tyrosine. Their phosphorylation actually went up. But not all the tyrosine phosphorylation cells go up. For example, this is a EGFR in the same cell. The tyrosine phosphorylation not change. So the ERB2 is the one change much. And going down to the downstream, the AKT pathway change, and also the P38. So the pathway, we can see the signal going down. Then one thing is also, which I told you in the morning, is the PAP, the cellular PAP, went down. ERB2 phosphorylation go up, cellular PAP activity going down. So that's what I show you the data in the morning is there's a strong inverse correlation between the PACP and the ERB2 phosphorylation. One went up, one go down. Then we show the data is PAP regulate ERB2. Okay, regulate ERB2. So this one is shows exactly the ERB2 is a key point of one. So then we further determine which site going further, because ERB2 has many tyrosine phosphorylation sites. So we try to identify which site is involved. Yeah, which site. Then will be more specific. So you can see here, it's actually, again, it's a tyrosine 1221/2. This one is actually changed. 1248, it may be changed, but may not be. Okay, in one cell line, L1 cell, we did not see exact the change, but in MDA, yes. So this is some sort of inconsistent. It's a cell line unique phenomenon. Okay, but then to another site, it's a PY 1112. Okay, 1112 position. There's no change. So we can, if from this data indicate the 1221/2 is the primary site, because it's a common phenomena as a phosphorylation going up in both androgen independent cell. So that's why how we are going to do the determination. If you have a computer, yes, the computer can tell you what could be the correlation, but we are the one ultimately pick what is really the key point, the functional. Okay. So this just show you the cell proliferation. Say this C81 cell indeed grow faster than the C33 cell and MDA cell, the AI cell, androgen independent, grow faster. So when the cell growth increase, become androgen independent, ERB2 phosphorylation at the tyrosine 1221, 1222, so that's 22, is a site correlated. Then the CPAP activity went down. Okay, so CPAP. So other site, actually other protein can also phosphorylate, but they are the downstream because ERB2 is on the plasma membrane, they're at the upstream. So we try to find this from upstream going down to the downstream. So that's what the significance is. We can see the whole systematically growth regulation. So that's what we are doing. Any question here? So this is a correlation in this moment, okay? So in here, then we actually use different kind of inhibitor. So the inhibitor is AG1479 or AG879. Because AG879, I remember that is an inhibitor to ERB2, and the AG1478, the first one is actually EGFR inhibitor. So we use a different inhibitor for the comparison. It really, so these two inhibitors are actually relative specific. So the EGFR inhibitor, the 1478, would not cross inhibit ERB2, would not. So similarly, the ERB2 inhibitor, A79, cannot inhibit EGFR. So luckily, so has been tested for a lot of people. We are not the only one. So that when we use inhibitor, we have confidence say this inhibition only to this specific kind receptor, but not the other one. So the data will be specific. So you can see the ERB2 inhibition greatly inhibits the cell proliferation. Okay, then the EGFR inhibitor actually have not much. Okay, so it can inhibit some sort of androgen stimulation, but not so dramatic as the ERB2 inhibitor. Not so dramatic. So that actually give our thinking is ERB2 could be the primary one, but not EGFR. But EGFR still can still can affect the growth, androgen stimulation of cell growth. The reason is EGFR and ERB2 can form heterodimer in growth regulation. They can form heterodimer. But the bottom line is, if ERB2 is the primary one, and we use that inhibitor, we can see more distinct separation, growth separation. Okay? But not as other one. Other one serve as a cofactor. So they do form the heterodimer in the cell. So this inhibitor experiment is something to give us a correlative result. Inhibition always correlated result. Then how can we do? At that time when we do the experiment, we actually have a special cell line. We have an antibody, the cell express antibody targeting to the ERB2. And then, interesting, this antibody have a link, could only localize, antibody can localize in the ER, cannot go to, so antibody will bind to the newly synthesized protein, ERB2 protein, retained in the ER, endoplasmic reticulum, but not going to the plasma membrane. So the functional one is on the plasma membrane, but not the ER one. Okay, so we can trap the ERB2 in the ER, endoplasmic reticulum, would not let it go to the plasma membrane. So they cannot function properly. So by this way, we find out it's actually androgen loss the stimulation. The wild type cell, the growth stimulation by androgen, but not the ERB2 trap one. So ERB2, they still express, but it's just been trapped in the ER, cannot go outside. So cannot functional properly. Then we lost the growth stimulation. So that shows ERB2 is involved in the androgen stimulation. Okay? Are we okay? Okay. So this is again is a lot of phosphorylation. We try to identify which phosphorylation site. And yeah. But the data here, the figure 2 is essentially to show ERB2 is a one, the molecule involved in androgen stimulation. So that is the one we trapped ERB2 inside the cell. Now what we are doing is we actually use ERB2 cDNA transfection, okay, ERB2 cDNA transfection to see the change. So after we transfect with the ERB2 cDNA, when they increase, the cell growth increase. Okay, so ERB2 indeed can stimulate cell growth increase. Then we actually on the other side, we knock down the PAP. So remember, we are just mentioned this PAP is ERB2 interaction, right? So if we inhibit the PAP, so ERB2 phosphorylation go up, right? Okay. If we knock out the phosphatase, the kinase will phosphorylate activity going down. So we use a cell, we knock it down by with the siRNA to knock down the phosphatase inside the cell. So when the phosphatase was knocked down, decreased, then the ERB2 phosphorylation going up in here. And again, the same site is the 1221/2, this one, but not 1248. Only this site is really the one shows. Yeah. And we also check other phosphatase, the SHIP1, SHIP2. So the SHIP1 we say mainly plays a role in B cell regulation. SHIP2 for the T cell. Then in the process cell, they also express these two proteins, but so when they go up, ERB2 phosphorylation go up. What does it mean? Can they function to interact with the ERB2? Could SHIP1, SHIP2 regulate ERB2 phosphorylation based on this data? Can it happen? If SHIP1 and the SHIP2 regulate ERB2 phosphorylation, so when the SHIP2 and SHIP1 going up, the phosphate going up, what happened to the ERB2 phosphorylation? Should go up or should go down? Go up. But the SHIP1, SHIP2 are the phosphatase. When they go up, ERB2 should go down or go up? Should go down. Because phosphate going up, the corresponding kinase should go down. So they don't follow the dogma. They don't follow the relationship. But only the PAP, it went down, it goes up. Okay, it goes up. So this is again a lot of data. When you analyze the data, how you can make the judgment call. People can get a lot of people can get a similar result, but who can make the judgment call accurately? This makes the difference. So we can knock down the phosphatase, the ERB2 phosphorylation going up, the cell proliferation also go up. So again, this is a difference. So we can express the cDNA, increase the cell growth. We can knock down the regular phosphatase, the ERB2 phosphorylation going up, the cell growth going up. So try to demonstrate a phenomena. Do we see the phenomena? We did not. We actually see the result. It's a correlation. So we need to perform different kind of correlation. The overlapping all in the circle, that is indeed is the case. Okay? So let's try to explain this. Yeah, we overexpression the kinase, cell growth increase. We knock down the corresponding phosphatase, the cell growth increase. So indicating this kinase involved in growth regulation as this phosphatase too. Yes? Any question? Okay. So because our purpose is going to demonstrate ERB2 phosphorylation involved in cell growth, in androgen responsive and also in castration or androgen independent cell. So this is a further study, more detailed study. We compare the ERB2 and the EGFR in the castration-resistant prostate cancer cell. So we can use, let me see. Okay, so we treat the cell with either the ERB2 inhibitor versus EGFR inhibitor, and to see the growth inhibition. Okay, so this is a treat with AG-A79, and this is a treat with AG-1479. So the growth inhibition only happening in the cell treated with ERB2 inhibitor, but not in the EGFR inhibitor. So it shows even the EGFR can interact with the ERB2 or form heterodimer, but ERB2 is the primary molecule responsible for cell growth. If we combine the two, the last line is the combination of the two. Yeah, we got the same inhibition, the same as the ERB2 inhibitor alone. So if EGFR inhibitor also have a function 1 + 1, we should obtain more than 1, right? But then in here we have 1 + 1 equal to 1, indicating the EGFR inhibitor really have no function in this way. Okay, no function. It's not involved in the androgen independent. So again, this is the one where we shows yeah, this is similar result. We actually doing the same. Okay. So the data just try to show is really the ERB2. Then also the Western here to show the inhibition. Let me see whether, oh yeah, I forgot to enlarge the figure, sorry. Yeah. What? Oh, yeah. I tried to see whether we can enlarge it, but it seems not. Oh, yeah, that is better. I forget that. I tried to, yeah. No, I did not. Sometimes it's when you stand up here, it totally mix up. It cannot see the computer well. Then my glasses always fall down. That's a problem. This is a no lineless multifocal issue. Yeah. So the bottom line here is we really try to see is how is the regulation is the ERB2 alone, or the EGFR also involved. So we try to have different approach back and forth. Use the cDNA, use the siRNA. We use a different inhibitor, and then for comparison at the same time. Okay, we need to do the experiment at the same in the same time. We cannot say, yeah, I do the inhibition here first 2 lane first, and then I do the second 2 lane first, and then put them the data together. Then the data may not fit. Okay, it's not good. So it's good is put all of them at the same time. Yes, it's a lot of things going on. You need to take care of them, and you need to run a lot of experiment, Western. But for the regular of the experiment, regulars of experiment, yes, that is the only way you need to do. We have no choice. So for time kinetic, for example, I even sleep in my office. When my son was only, my daughter was 1 year old, 3 months old. And I need to have my wife's understanding. I sleep in the office. because the time kinetic, I need to harvest the cell for experiment immediately, every 3 hours or 6 hours. So she need to stay home taking care of the baby with our very active son. Okay, so this is for experiment, and at the early stage, you really need to spend a lot of effort. But on the other side of the coin, when you got it, then you really feel very, very exciting. So this is other side is not only doing the cell growth. So the cell growth actually is one kind of the biological effect, yes. But this is agar emerging. It's in the soft agar. So what is soft agar? Yeah, the soft agar is actually, we call the cheaper experiment to perform tumorigenicity, the cheap assay. So that means the normal cell, they cannot grow in the soft agar. It's a 0.3% agar loss. Okay, 0.3% agar. So a normal cell, they cannot grow on the top of 0.3% agar. Only the cancer cell. Why? Because it's a soft. So the cancer cell don't have anchorage dependent cell growth. The normal cell need to have anchorage dependent. So this is one of the criteria for cancer cell. They have anchorage independence. They can grow suspendedly. So we can see with the inhibitor, this AG-A79 is ERB2 inhibitor. Then the cancer cells, they grow well, a lot of colonies. But when with the ERB2 inhibitor, the colony number going down and smaller. But with the EGR inhibitor, 1479, they still very big size. So the number a little bit less, but the size actually very similar, no change. So they combine the two, the number decrease, the size slow down. So again, the data shows the ERB2 not only involved in cell growth, they're also involved in the tumorigenicity, So this is a content. We analyze whether it's a significant difference. Okay, so we need to count the colony, how many of them under the microscope, and then we need to, of course, perform several set of experiments and then have a mean of it. So in here we show is experiment n equal to 3. So that's what I mean is we need here n equal to 3 is we at least have 3 dish, petri dish, 3 of them, and we count all of them, and then we got the average here for statistics. So we use n equal to 3 usually speaking. Then the p-value usually need to less than 0.05. What the meaning p less than 0.05? What does that mean? Yes, significance of the data, but what does that mean? So p less than 0.05. What does that mean in the p-value, in the statistic? No, it's significant or non-significant, but what is the meaning beyond that? So that means over 95% of the experiment follows the same. Okay, or over 95% of experiment result, they follow the same trend. So that means over 95% of the cell of the experiment, the cell growth, the soft agar growth will be inhibited by ERB2 inhibitor, but not by EGF receptor inhibitor. So then less than 0.01, that means over 99% get the same result. And usually we cut off at 0.05 because 95% follow the rule. So that is pretty significant, 95%. Then p less than 0.01 even much significant because 99% following that pathway. Okay, so that the p-value means the meaning of the p-value. Any question? Okay. Yeah, so this is a scheme we show at the time is the androgen dependent cell. When without the androgen, the PAP is actually high because we show with the androgen, PAP go down, right? The phosphate go down. So without the androgen, the high PAP phosphate ERB2. Then with the androgen, the PAP going down, the ERB2 phosphorylation going up, then they go down to the stream, the P38 or the ERK pathway. Okay, the trend. So the ERB2 can have a signal going down to the stream to see the growth regulation. But in the androgen independent cell, the phosphate actually decrease. That we show the data, the C81 cell, the androgen independent cell, the phosphate decrease. ERB2 phosphorylation actually going up, right? So as a set of tyrosine 1221 and the 1222. So those are two tyrosine adjacent to each other. They both will be phosphorylated. Okay. Then the antibody can recognize both tyrosine phosphorylation at the same time. So then in the androgen independent cell, even because the phosphatase going down decrease, so ERB2 is hyperphosphorylated, then they transduce the signal going down, the cell growth going up. The PSA is a marker, as I mentioned, it's a prostate cancer marker. Then why in the prostate cancer, the PSA going up? Because we have other publication shows ERB2 can upregulate PSA expression. Okay, ERB2, the tyrosine phosphorylation, can indirectly through the pathway to activate the androgen receptor. Then the androgen receptor can turn on the PSA secretion. So why in the advanced stage of prostate cancer, we can see the PSA increase in patient circulation, and why the PSA, the serum marker PSA, can serve as a marker to detect prostate cancer. Because in the cancer cell, the phosphate is actually inside the cell, the phosphate going down, the ERB2 phosphorylation going up, then they can go to activate the PSA expression and go to the circulation. Okay, so this is how we are going to perform the experiment. We are going to demonstrate the experiment back and forth. We use an inhibitor, different inhibitor for comparison. We use the cDNA expression, going up the expression. We use siRNA to knock it down. And we even use the cell line. We can trap the ERB2, retain intracellular in the ER, will not go to the membrane, so have no function. So all this together shows without the ERB2, then androgen stimulation abolished, androgen independent is gone. Then with, okay, so indicating the ERB2 is a key player, but not the EGFR. Are we clear? Okay, so this is where when you are going to publish a paper in a solid journal, cellular signaling is not a very high impact journal, but it's a very solid journal. It's a very solid journal. They review very carefully. And again, it's whether the data really support the conclusion. Very solid journal. As one of my colleagues, he published his paper in the same journal. He said, yeah, you should be very proud. You can publish it. Even it's not high impact, but it's a solid journal. As yesterday, I remember one student to ask me whether we need to focus on the high impact journal. Of course, if possible, we go to impact journal. Not only it's a lot of factors we need to consider whether you have time to fight with them, because they're always asking for a lot of additional experiments. So whether you have time to answer to it, and whether you submit, you revise it, they will sometimes will still come up with a new additional comment. You need to revise it again. So if you are a student, can you afford to wait until your paper published before your graduation? So if the answer is no, then we choose will be the solid journal. Okay, the solid journal. it will be very important. Then that will actually, especially those traditional journals, that actually they give us a lot of feedback, a lot of comments. So this is one how we perform. We can demonstrate the interaction of the two proteins involved in the regulation, androgen regulation. So another article, any question, or otherwise I might try to go through. Next one is to show is the JBC paper, how we yeah, sure.
Earlier today, the cell density
Yeah.
correlation with androgen.
So I
was wondering, since the cell can sense cell density, so could we say that it is a form of contact inhibition? If not contact inhibition, then could we target PAP or related proteins to induce contact inhibition to slow down metastasis?
it's a very good question. Is normal cell have contact inhibition, but not the cancer cell. The cancer cell lost contact inhibition, so they can grow in the pretreated, for example, multi-layers, and they can form a solid tumor because they lost contact inhibition. Okay, so whether we can reduce contact inhibition, say we actually should restore their contact inhibition, then to inhibit their growth. Yes, we can do that, but the contact inhibition involved in a lot of factors. So it will be a really big challenge. Of course, that involved in the cytoskeleton protein, involved in a lot of actins. Yeah, it's a lot. So yes, if we can increase contact inhibition, we should say if we can restore contact inhibition, of course they go back to the normal phenotype. But it's just a tough challenge. Okay, a tough challenge. Okay, any other question? Yeah, so that is a show, that the data shows ERB2 plays a critical role in the androgen stimulation in the advanced stage castration-resistant prostate cancer cells. So ERB2 targeting is important. But then the problem is, I actually had just come up to my mind, is actually a question we were asking, I was asked before, is then if ERB2 is involved, can we use a Herceptin targeting to ERB2? Can we use Herceptin to inhibit prostate cancer cell? How do you think? Can we use a Herceptin? It's ERB2. Actually, it's a ERB2 binding antibody. Okay, to inhibit the prostate cancer cell. Can we? No, we cannot, because HER2, Herceptin only recognize, amplify ERB2. When the ERB2 molecule going up more molecule of ERB2, then the Herceptin could recognize it and could inhibit it. But in the prostate cancer cell, the protein level no change. It's a phosphorylation going up. Okay, it's a phosphorylation going up. Therefore, Herceptin has no efficacy in prostate cancer therapy. So it's the same molecule. We need to understand is elevate amplification, elevate expression, or it is activation. So activation then. So now come back is if activation is involved, can we use the AGA79 to inhibit it for chemotherapy? Can we use it AGA79 for chemotherapy, for prostate cancer therapy? Unfortunately, the answer is still no. It's not to say it's the function is wrong. The problem is AGA79 in the circulation has very short life. Has very short life. So therefore, we cannot reach to the efficacy concentration. We cannot. So the short life. So they are actually trying to find a new compound can prolong the compound lifetime, then we can use it. Okay, so the compound can be used as a chemo reagent or cannot. it's not only the specificity, but also we need to test whether what is the half-life, what is the lifespan of the compound in the circulation. You're too short, say, you intake it, the compound hydrolyzes immediately. Then even before they reach to the cancer cell, how can they function? So for a compound, we need to test for their half-life, at least a beginning in the animal. Then subsequently, we need to put it in human being too. We need to do the kinetic study to understand how long they will survive. Okay. So if a prescription, for example, in the AIDS virus, at the beginning, all the patient need to have the prescription, the compound reagent every day, daily for the AIDS virus. But now they develop the prolonged release compound, or the slow, they can stay in the circulation for a long time. So now for the AIDS patient, they might have some kind of injection once per month, once per 6 months. Because it's a long life reagent. So that will reduce the patient's burden. So the patient does not need to go to clinic, get injection or get a prescription all the time. Okay? Yeah, so a compound development, a drug development, we need to have many considerations. Not only the specificity to reduce the adverse effect, we need to consider the kinetic, how long they can survive in the circulation. So that actually could reduce patient's burden to have the drug continuously. Then if the kinetic, okay, is not good, they have short life, they may not be able to use, like ERV2 inhibitor, AGA79. And not only that, ERK inhibitor. So we observe the inhibition, the pathway going down to ERK, right? So ERK actually serves as a louder pathway, a merge of the louder pathway, okay, ERK. And ERK is actually activated in the louder cancer cell too. So initially, we did propose, as other people, is whether we can use the ERK inhibitor to inhibit the cancer cell. And unfortunately, the ERK inhibitor has very short life in circulation, very unstable. So we can do it in the experiment, but not for clinical research, for clinical usage. Okay. Yeah, because I remember most of you are the graduate student doing experiments. So these are the some sort of information you are involved in the drug development. Then you should recognize, understand how much, what kind of which aspect you need to pay attention to. When you have a compound, one is specificity, one is at least the half-life. Okay, so the toxicity, the specificity will reduce the toxicity, will reduce the adverse effect. But the half-life is too short, it cannot use either. So for example, very simple, cyanide. We know cyanide, right? GIAN, cyanide. It's very toxic. So the idea is, can we use a cyanide to kill cancer cells? Can we? Yeah, it's very tough because it's very toxic. And not only that, we need to consider its safe window, the efficacy window, how much it gets efficacy and how much they will become the toxic. So we call this a safe window. So like Tylenol, you know the Tylenol. I don't. You use the same term, right? Tylenol, to painkiller Tylenol. The Tylenol, why you can easily purchase it from the counter, over the counter? Why? Because the safe window is huge. So even the recommendation, you had say 1,000 milligrams. Even you had 2,000 milligrams, still okay. So when the cell window is huge, then it can be as an over-counter drug. But if the huge, the cell window is very tiny, like those cancer drug, it's very toxic. So the administration need to be very careful. Then in that case, all the IV injection, you need to go back to clinical. You cannot do it at your home because cell window, we call the cell window, is very narrow. So cyanide is actually being used in the Chinese medicine for some disease. So for example, you got a fever, the cyanide can actually use it to release, to have the fever release. But very few Chinese medicine doctor is willing to give it this prescription because very tedious. The cell window is so narrow. Okay, you give a little bit more amount, it might cure the patient. Yes, we can use for curing. So there is a lot of compound we can cure the cancer cell, but it's just unfortunate too toxic. The cell window is very small, so very difficult to administrate the compound. Any question? Yeah.
The cancer cells have high Hayflick limit, like they grow in exponential form. Can we not use any inhibitor or something to reduce their Hayflick limits?
To reduce?
Their Hayflick limits. Hayflick limits.
I didn't know. What chemistry?
We know that there are telomerase and certain things that help the cells to divide.
Yes.
But in cancer cells, these telomerase are in high number. That the Hayflick limit is in exponential form, and the cancer cells grow and grow in very large amount. So can we not reduce their Hayflick limit or telomerase to slow it down, and then we can control them longer?
So the cancer cell growth increase actually, they do have different pathway. Okay, so how we can reduce their cell growth actually is a lot of study going on. Even now, metabolic analysis are involved in a lot because they actually change the metabolic regulation in cancer cell. I did not mention it clearly. The metabolic, so for example, cancer cells use a lot of glutamine for their TCA cycle. They actually change their metabolic regulation, but not the normal cell. So when we culture the cancer cell, sometimes we add additional glutamine to help their growth, but not in the normal cell. So they change the metabolic regulation. So for the last few years, actually, metabolic regulation become a very hot topic. That is what they try to modify their regulation, their metabolic regulation for the growth regulation. So in the free radical, for example, again, if we can reduce the free radical, we can inhibit the cancer cell. So yes, but how we are going to target it, which enzyme? So I think, yeah, Friday I was talking about that. For example, free radical antioxidant is actually have a very major clinical trial, and it turned out in the clinical trial was not successful. Okay, antioxidant actually increase the cancer cell growth, but not reduce. So I think I did mention yesterday is whether we give the patient not early enough, but too late. We should give them earlier when they are at, say, age 20s, 30s. But how can we give a person at young age so much amount of antioxidant what could be the long-term effect? I don't know. We don't know. So there is a lot of unknown factor going on. So yeah, so tomorrow is Thursday, right? Yeah, Friday. So they switch my talk. So that's the topic. Yeah, on Friday, in the Friday morning, I'm going to mention about say the targeting is antioxidant targeting for the treatment, but it's just failure. Okay. So again, it's whether we can develop specific one targeting to a specific growth regulation, either in the redox pathway or in the hormone pathway. Yeah, so that actually is a possibility. I can tell you, so even they try to develop the compound, because we know in the mitochondria, the charge different. There's a lot of electron change in the mitochondria, right? It's a respiratory chain. So some people did develop some kind of compound. They have different charge. They can activate the compound when they get into the mitochondria. And we know the mitochondria in cancer cell more active. So they use a compound can be activated when they move into the mitochondria for the therapeutic purpose. But turn out the result was not good either. Yeah, it has been 15 years ago already. I remember the idea came out about 15 years ago. But it just, so there's a lot of results, there's a lot of concept, and we will all try the best we can, how we can target it. But yes, they do make the progress, okay? But it's just very slow, just very, very slow. Yeah, but in overall speaking, yes, the cancer deaths actually go down in some cancer, in part based on the early detection. So the best way is early detection, then you can save the patient's life better. Okay, so the tumor marker now still a very, very hard area for the study. Even not the tumor marker, any marker, like Alzheimer's disease, we all go to up, I go to all. A lot of people will get the problem, right? So can we have a biomarker can predict the patient progression to the Alzheimer's disease? Okay, so those are a lot of study going on in every single aspect. Some of them you never ever think about, but they are doing it. Yeah. Okay. So sorry, I cannot give you all the critical, clear answer what it is. So this is actually, I think I will quickly go through it. This again, they show you how we determine the protein-protein interaction. Okay, how we determine ERB2 is actually regulated by the phosphatase, the PACP. How we determine it. So the first again is we have different cell line, and we show the inverse correlation. It's shown in this figure. Say the CAT1 cell lost the PAP, as I told you already. Then the ERB2 phosphorylation going up, then essentially it's a 12, 21, 22, but not the other side. So very specifically, tyrosine at the 12, 21, and the 22 actually inverse correlate with the PAP activity. Not only in RNCP cell, but also in the MDA cell. When in the passage 81 is going down, phosphorylation going up. Okay, so we can show this phenomena in at least two independent cell lines. And the cell growth also increase. When the PAP decrease, ERB2 phosphorylation going up, the cell growth increase. So again, this is just we show ERB2 phosphorylation correlated with cell growth. So then we try to determine whether they interaction. So the best way to see whether they can form the complex. When they interaction, they should form the complex, right? So we can do the co-immunoprecipitation. So we appreciate one to see the another one, co-immunoprecipitation. Okay. So then we immunoprecipitate phosphatase, then you can see the ERB2 co-precipitate. Then conversely, we can precipitate ERB2. We can see the phosphatase co-IP, co-precipitate. Then this is again is a where we try. Okay, so here, then this is where it is interaction. Where is the ERB2? Where is the PAP? Turn out they are actually working in the cytosol area. So we subfractionate the cell into the membrane, cytosol, and the nucleus, and we find the phosphatase actually going down. They can be in the cytosol, mainly in the membrane. They can be in the cytosol. Some of them even in the nucleus. Some of them even in the nucleus. And the interesting, the ERB2 distribution in the circumference cell and the circumference cell, some sort of different. So cell density effect, where is the protein? Okay, so we quantify and we have the percentage, and then we also analyze the phosphatase, where it is. Then this actually is that really the cytosol fraction or the nucleus fraction. This is a marker for it. So the bottom line is the PAP and ERB2 can co-precipitate, co-IP. So indicating they can interact. They are in the same complex. So whether the complex has another cofactor as a bridge point protein or something, at this moment we cannot tell. We don't know. But at least we know they are in the same complex. So we can precipitate one, we observe the other one, vice versa. So shows the two form the complex. Okay. And the PAP actually interact with the ERB2 actually can be in the membrane fraction, mainly in the cytosol, and only the mature form. PAP has immature form is a 42 kilodalton. Only the mature form can interact, can co-IP. So this actually can serve as a control. Say, immature form, inactive form cannot in the complex, but only the active form can be in the same complex. Okay, and then their interaction actually primarily actually in the cytosol higher than in the membrane. So that actually indicate the phosphatase is a cytosolic protein. Yeah. So again, is that really the case? It's really PAP regulate ERB2 phosphorylation. So, and then it's the PAP activity, the phosphatase activity involved. So what we did is we perform the pointer mutation on the phosphatase. We have the mutation at the H12 position to alanine. We have Y type. So this is the inactive form of the phosphatase. Then we can see we express it. Okay. There's the same amount of the protein, but then the function 12, 21, 22 decrease mainly in the active, the wild type one, the active. So this is a wild type PAP, and this is a mutant PAP. So the wild type, only the wild type PAP expression, the phosphorylation going down. Yeah, the phosphorylation going down. So here, unfortunately, this data was not so beautiful. But at least it clearly shows that is the case. Even in the 48 hour and the 72 hour shows the same, the similar. So the phosphorylation of ERB2 actually is dephosphorylated by the active form of the PACP. Then the inactive form of the PACP, the H12A it actually have no function. So this indicates it's not really they form the same complex. The activity is involved in the dephosphorylation. Okay, so again, it's how we determine it, how we do it, and then not only the two, if their interaction, can they change, alter the downstream signal? So the Shik is the downstream of the ERB2, and then PCNA is the cell growth. We can see this dephosphorylation modify, alter, regulate the downstream molecular signal. So it's a functional effect. Sometimes you do the dephosphorylation, but we cannot see the functional effect. So that means this regulation is not biological significant. But now we can see the dephosphorylation correlated with the downstream signal change. So indicate it's biological significant. Okay, so when we do an experiment, we need to be very careful to analyze the biological relevance. So overall, in this one, we actually knock down the phosphatase or overexpress the phosphatase. Okay, this is expressed phosphatase by cDNA. When expressed PAP, the phosphorylation going down. Yeah, so this is a control cell. So again, we knock down the use the siRNA, we knock down from the dosage, then PAP decrease, the ERB2 phosphorylation going down. Yeah. So then cell growth also going down. So the PAP ERB2 interaction actually indeed regulate cell proliferation. So this experiment, when you in your research, okay, what you need to do is what I explained to you is you need to use different approach, different angle. Only the cDNA expression, it could be artifact. So then you need to overexpression it or knock it down to see whether the result opposite. If then overexpression result opposite to the knockdown result, then indicate yes, that indeed the case. Okay, then we can use the cDNA for overexpression. We can knock down with sRNA or shRNA, or we can use the inhibitor. We can use the active form of inhibitor. We can use inactive form inhibitor. They do have inhibitor, active inhibitor or inactive inhibitor as a control. So we need to use a different approach. So that is what a solid journal, what they are required. Okay, a solid journal. So again, we do the time kinetic. Okay, so that was the dosage, different amount of the siRNA. And this is the kinetic. We transfer with the knock down the phosphatase and the following different time to see its change. To see its change. So for biochemistry, as I did mention, is the kinetic and the dosage are the two gold standard. Okay, you do the kinetic, you do the dosage, the two will match, then it's really, we can really believe what the data counts. Yeah. Then this is, so the previous one was not is a mixed cell line, the mixed cell which contract with siRNA. So now we establish a subclone cell line, and to further establish whether indeed that is the case. So we can knock it down the phosphatase with the subclone cell line, and then the ERB2 phosphorylation going down. And this actually correlates with the cell proliferation. Okay. So the phosphate going down, the cell growth increase over and over. And not only that, they develop tumor much faster and much bigger in the mouse. So this is knockdown subclone cell line C11, C3, C7, and this V3 is a control cell. So not only affect the cell growth, but also affect the tumor genesis in the mouse. So they develop tumor when there's no phosphatase. They develop tumor in the mouse even much faster, bigger size. Okay, so I think that what I try to say is, I show you two publications from us about how the ERB2 PAP interaction and how their functional role. The key is really, for a study journal, you need to do is more than one cell line. And your approach is more than one, not only the inhibitor alone. The inhibitor alone now, essentially, it cannot go through a study journal. Okay, you need to have different approach. Use the siRNA to knock it down, use the cDNA to express it, express it, use a different inhibitor. So even we test androgen affect calcium mobilization, we use 3 different inhibitors to PKC to inhibit the activity. And we even use a positive TPA to show indeed TPA and androgen could stimulate cell growth, to stimulate PKC activity similarly. So all this put together to tell you what to be a rigorous publication. So for grant review, usually when manuscript review, usually that's what we are checking is, what is your N number? Your N number, if you did not show your N number, we don't know how many times you did it, how good the p-value. So it's impossible to be accepted. Okay, so the paper discussion this hour is just I try to reiterate how we do the experiment and give you the example, give you some idea what's going on. The key is you need to back and forth, whatever you could, different approach, whatever you can think. Even we did not think, we have cell line, we chop the ERB2, retain in the ER, and lost activity and loss of function. Yeah, this is very incredible. Actually, it was developed by other lab, and luckily we can get the cell line for the experiment. And this in addition to the inhibitor, in addition to the knockdown or the cDNA expression. Okay, any question? Any experiment question, scientific question? Are we okay? Yeah.
Young man, we are injecting estrogen by injection.
Injection estrogen.
Injecting estrogen by injection. Then what is the effect of androgen cycle on that person?
If a young man injects estrogen,
2 months regularly we are using that.
a young man,
Male,
young male injects estrogen.
estrogen we are injecting by injection, approx 2 months. Then what is the effect of androgen? How signaling affect androgen signaling?
The androgen signal will go down, and the male will become a female. Yeah.
Really?
Yes. When you continue injection, inject 2 months, I don't know how much effect, but you continue injection. So there's a sexual conversion. That's what they are doing. They inject this. Say your male try to into the female. Yes, one is we need to remove the male organ. Okay? Then another is to inject with the female hormone. Yeah.
Another question. This is general question coming in my mind.
Yeah.
So we are generally using goat and chicken we cut and eat as a food. There is no ethical issue. But we are using a practical for research. Even if you are using a small mice, there is a ethical issue. Why it's happening?
Oh, when you consume the food, whether there's no ethics issue, yes, they do. Oh, yes. So the USDA, for example, in USDA is the one they are responsible for regulate this kind in the animal, okay? For example, how they are going to sacrifice an animal they have very strong regulation, very tight regulation. For example, they use electric shock to kill a pig. It got to kill the pig, they cannot let the pig suffer it. Oh yes, they do. And the same, they actually killed. I know the pig better because my father is a veterinarian. Okay, so he's an animal doctor, studied the vaccine. So yes, they do have their regulation. They are on. Just we don't know. That's all. So yeah, we cannot let the animal suffer before we sacrifice. No, we cannot. They need to be sacrificed ASAP. If we are going to sacrifice it, we cannot let them scare. Actually, animal has. Yes, I can tell you. Actually, the animal have that kind of feeling. So we cannot let them suffer. So that we torture them, let them be under the stress over there. No, we cannot. So they do have their guideline. They need to sacrifice them within certain time point. So don't complain about your experiment, okay? Yes, in USDA, that in US, we do have very tight regulation. Yeah. So like the cow, the pig, they are going to be sacrificed. They need to use a high voltage electric shock. They will pass away within seconds, so they will not suffer. And we cannot put them in the small cage. So the guideline is also, for example, we cannot maintain them in a small cage for how long? Okay, so say like a chicken, they do have a regulation as we are in the lab. How many mice in one cage, right? So 4 mice or 5 mice in the regular cage. So they do have their guideline. How many chicken in how many square feet? They do. Cannot be too crowded. But during the shipping, say before they are sacrificed, maybe more crowded, yes. We have no choice. Okay? But in general speaking, they do have their own guideline. Yeah. We need to treat them humanely as much as we could, even we consume their meat. Yeah. Okay? Any other question? Okay, I think, yeah. In that case, then we will finish today, and then I think we finish today, right? Then tomorrow we are talking about what? Yeah, the cancer target and cancer treatment. Okay. Then finally, tomorrow actually initial the syllabus will be I give 3 hours and then finish it, but I just been told by Dr. Kumar is I will give 2 hours, then 1 hour will be from outside speaker talking about his research related one. Then my last moment to tell you how we perform our interaction of the signal transduction. I mean how the steroid hormone, the free radical, they integrate into the growth regulation. Use the P66 chick as an example. I will present on Friday morning. Okay, so there's a little bit of schedule change. I think you guys have that one updated one. I did not have updated one until this morning. I was told. Okay. Okay. Any questions? Send me an email. I will try to reply. I know one of the students sent me the email requesting for the slide. Unfortunately, Monday night I was so exhausted and I did not get and actually the email did not arrive. Tuesday morning when I came here. Yeah, that's what I went back last night. I saw the email, but yesterday morning I did not get the Monday night email. So sometimes the email just they like have troubling or troubling for fun. Okay, you really don't know. So for example, my office and the department office are just 2 different buildings, not far away. Then the secretary sent me the email. I might wait for 1 hour to get it. Yeah, I might wait for over 1 hour to receive the email from other building in the same campus, in the same system. Yeah. And sometimes, of course, very fast, but sometimes it happens. So the email just have their own choice. They like to wandering around. Okay, thank you. Yeah, then see you guys tomorrow. Yeah. Thank you. Yeah, bye-bye.